Dr. Bert Calleweart is a geneticist known for researching rare genetic syndromes. His dedication to understanding the underlying genetic mechanisms of conditions has contributed to advancements in genetic diagnostics and therapies, and is here today to update you on the Myhre Zebrafish model. Dr. Calleweart, we're keen to hear your updates, so over to you.
Okay, thank you so much for having me on this wonderful symposium. It's really a pleasure being here with you all. Good afternoon in the United States, and good evening and those that are in Europe. So I would like to update you on Myhre syndrome research. So if we diagnose a child with a genetic disease, whether it be Myhre syndrome or another genetically determined disease, we will of course search for the underlying gene defect. But then we are still far away from therapy. So to understand which therapies might be useful, we have to know the mechanisms that actually cause the disease. So the links between the gene defect and the actual symptoms of the disease. And therefore, we often need animal models. And in our lab, we use both fly models, zebrafish models, and also mice models. But I would like to update you on mice models, as these have their specific particularities that are especially of interest also in Myhre syndrome. So we have these new tools that have been developed over the past years that's called CRISPR-Cas9 mediated genome editing. With this technique, we can actually quite precisely edit the genome of a model of an animal model at a certain spot in the DNA. So we can specifically introduce variants that cause disease. And we like to work with zebrafish in our lab. Why do we actually choose zebrafish? Well, I think one of the main reasons is that you can see over here, well, we can see the development of these zebrafish throughout the larval stadium at 72 hours. So we can actually see every cell division in the growing embryo. And these embryos are transparent at least up to 48 hours, so we can see everything quite in detail under the microscope. We can see the vertebral column, we can see the eyes, we can see the heart developed, and we can also see the blood vessels. Moreover, it's a relatively cheap model to keep. They have high fecundity, so we have up to 300 embryos a week from one single breeding pair. As said, they are transparent and they have the external development, and there's quite a large conservation between the human genome and the zebrafish genome, up to 70%. Also, we can kind of modify the zebrafish in order to express fluorescent cell markers, meaning that, for instance, we can enlight the endothelial cells on the vasculature, making sure that we can very nicely see a fluorescent image of the vessels that are developing. And finally, also, zebrafish have this specific capability to regenerate their tissue. So this might be a very interesting model to specifically look at scar formation and fibrosis, which is one of the major issues in Myhre syndrome. And then finally, because of the high fecundity of these animals, we can also do compound screening. So we can test whole batteries of known chemical compounds and see if any of these compounds may actually change the natural disease course in these fish. Of course, any model also has some disadvantages. And for zebrafish specifically, that is mainly the genome duplication they had in the past. Like every fish, they actually have duplicated their genome, meaning that they often have 2 copies for each gene, while humans only have 1 copy. And in zebrafish, this is often denominated the A and the B copy. Therefore, they also have more compensatory mechanisms avoiding disease manifestations. So not every model is always very successful in expressing the disease. Of course, they're not mammal, their sexual development is quite different, and they don't have lungs. They do have another structure that is embryologically related to the lungs, which is the swimming bladder. So a little bit more to understand the specific CRISPR-Cas9 mediated genome editing. This was a technique that was described by Jennifer Doudna and by Emmanuel Charpentier, who are actually microbiologists. And so to understand specifically this technique, you have to know somewhat on the background of how our DNA functions. So our DNA is actually a double helical structure. And so both strands are actually fully complementary to each other. And the DNA is built out of 4 different building bricks, C, A, G, and T. And in those 2 strands, T always complements A, G always complements C, and vice versa. So in order to make a kind of protein, a certain gene is being transcribed first to a specific molecule, which is called an RNA, and that's actually an exact copy of a specific gene in our DNA. This copy is then used by our protein-making machinery to make a specific protein. And this machinery actually reads this RNA in triplets of building bricks. So meaning that every triplet of building bricks encodes a building brick for the protein, an amino acid. So it's very important that this triplet structure is kept throughout the sequence. If just one building brick would miss in this RNA or in the DNA, then you get a totally other protein that is usually broken down. So what were we doing if we want to make these animal models with CRISPR-Cas9 genome editing? We actually develop a kind of guide RNA. So this is a molecule that recognizes a specific sequence in the DNA. And this guide RNA also brings a protein to the DNA, the Cas9 protein. And this is actually just a molecular scissor. When it binds to the DNA, it just cleaves it, both strands at once. And so once this is cleaved, the cell will try to repair this DNA. And then there are 2 ways. Either they will just try to paste both strands back together, and this is quite error prone. Often this results in the loss or addition of a few of these building bricks in the DNA. And as a result, if this happens, there's often a change in the number of, a change in the triplet structure, so that the protein is misassembled completely and broken down. On the other side, we can also add a repair template that helps the cell to repair it in a specific way. And if we add a certain change in the repair template, then we can actually change one of these building bricks rather than removing one, causing also a different protein structure. And this is actually what we need to do in Myhre syndrome, because in Myhre syndrome, the SMAD4 mutation is a specific variant that is one building brick that changes in the eventual protein. So what are the different steps that we have to do? Well, we have to inject all the components that are necessary in the fertilized egg. This will result in genome editing in some of the cells of the fish. So we grow this fish to adulthood, and some of these cells will have the desired change. Other cells will have other changes that we are not interested in, or no change at all. So after 3 months, these fish are adult fish, and we can mate them just with wild type fish that have no genome editing. And so this will cause a different number of embryos, all with different changes, but not mosaic anymore. It's present in every cell at that time. So then we sequence these specific embryos and see if some of the embryos might have the desired change that we want. Then again, we have to grow them into adulthood, mate them again with wild-type fish, and then eventually we can, after another round of growing them to adulthood, mate them again in order to reduce the number of unwanted side effects. For Myhre syndrome specifically, we know that this protein is also expressed in the embryo very early on, and this is still maternal expression. So this is still maternal protein that is present. So if we want to see very early phenotypes, you actually have to have affected females in the breeding pair. So this is quite a lot of steps, and this takes up to 9 months to 1 year at least to get a good model. And here are some pictures of our zebrafish facilities. So you can see the different tanks with all the fish in them. And this is our injection microscope. So this is actually a very fine needle that we use to inject the eggs, the zebrafish eggs, with all the necessary components. So if it's so hard to make all these zebrafish models, so why would we actually bother? Well, I can give you a nice example of another model that we have recently developed. The zebrafish model is currently under development. We have our adult fish that have been backcrossed already and that are now ready for phenotyping. But this will be the work over the next year to have a more clear idea of what the phenotype of the zebrafish is for Myhre syndrome. But just to give you a sense of feeling what we can achieve with these fish, I would like to present a short update on another project that was done by Michiel van Hooydonk, one of the PhD students in my lab. And he actually worked on thoracic aortic dissection. So this is a disease where a rupture may occur in the aorta, the principal vessel in the body. And this causes actually blood to flow into the wall or even rupture throughout the aorta, which is, of course, a very devastating disease, can be deadly if not treated promptly. Usually there is an aneurysm, a widening of the aorta just in the years before the rupture, but this is asymptomatic. So it's always important to identify these aneurysms in order to treat them in a preventive way rather than waiting until it ruptures. So we were actually interested in two signaling pathways, the TGF-beta and DMP signaling pathways, because they both signal through a same molecule, and that is the SMAD4 molecule that is implicated in Myhre syndrome. So we actually thought if we make changes in both pathways, what might actually happen? And the rationale for this was that if we look at SMAD3 and SMAD6, SMAD3 is in the TGF-beta signaling pathway, SMAD6 is in the DMP signaling pathway, we actually saw that on the chromosomes, so they are actually very close to each other on the DNA, both in humans as in mice, but also in the zebrafish. And the zebrafish, as said, has for each gene 2 copies. We know that mutations in SMAD3 cause a combination of skeletal problems with aortic aneurysms and some craniofacial characteristics, while SMAD6 loss of function can either cause defects of the aortic valve with aortic root dilatation or craniosynostosis, which is a premature closure of the sutures of the bones in the head. So we actually made models for all these genes, but unfortunately we didn't observe any phenotype in these fish. So we then went on by making double knockout zebrafish models, knocking out both the 3A6A, 3A3B, 6A6B, and 3B6B. And we observed that one type, the fish that lacked both the 6A and 6B, died prematurely. And this is what we could actually do to phenotype them. So we can actually see the vessels, and this is the main vessel coming out of the heart in the zebrafish that will bring the blood to the gills. And what we actually could see is that this vessel was much smaller in these fish. So we then went on with the next step, making quadruple knockout fish, deleting all these genes in the fish. And then we observed that many of these adult fish suddenly died, especially if we scoop them out of the water for 30 seconds, which is called a kind of stress test that is harmless in healthy fish, but this caused instant death in many of these mutant fish. And so they also had a lower survival rate. So what we then did is we actually imaged the aorta by making sections throughout the fish and compiling all these sections back together so we could have a 3D image of the ventral aorta of these fish. And this is then what you can actually nicely see, and you can see that here in purple, the dissection is clearly indicated. These are just histological sections where you can see this nice lamellar buildup of the wall of the aorta, of elastic vessels, and in between the smooth muscle cells. And here you can see a fish where there was a rupture and blood coming into the aortic wall. And this nice reconstructions can actually tell us what is happening, why these fish are dying, and what the initial changes are in the aorta of these fish. You can also very nicely see the imaging of the vessels, so we can mark these vessels fluorescently. And at 10 days, you can even see that some of these vessels were abnormally shaped with some huge tortuosity that you could observe, which is something that you also see in patients with SMAD3 defects. And in adult fish, we can even do echocardiography of these fish and look at the function of the heart, both systolic and diastolic function. So what becomes more interesting then is once you have a clear phenotype, you can search for the mechanisms underlying this phenotype. And for that, we actually look at the copies of the DNA that are made, these RNA copies. So this tells us how much certain genes are being used at a certain, in a certain tissue at a certain time point in development. And this is what we call RNA sequencing. It gives us a little bit about functionality of the DNA, both in wild type and in the mutant fish. And so what we observed is that genes that are implicated in protein breakdown were actually downregulated in these fish. So they were less active, these genes, so there was less protein breakdown in the model. Another thing we observed is that melanogenesis or pigment deposition was upregulated in these fish. And this is something novel that hasn't been identified in other models of aortic aneurysms. However, there are melanocytes, pigment cells in the aorta, so they must have some kind of function. And what is even more interesting is that one of the major genes that was upregulated, MSF, is important for pigmentation, but is also important for BMP signaling, which is also driven by SMAD4. So in future perspectives, we will do compound screening and look if we can ameliorate the phenotype. We will also do hemodynamical studies to understand why certain spots are more prone to dissection in the fish, and this might be able, we might be able to extrapolate this to the human diseases. So what about Myhre syndrome? So we have our zebrafish model. You can actually see here a fluorescent image of the vasculature of one of our first adult fish with Myhre syndrome. We're still doing the analysis, so I cannot say too much yet about this. And this is actually the work of Eva, who is also present here at the meeting. She will work for the next 4 years on this project. We can also do wound healing studies. So just by a laser, you can actually make a small wound on the skin of the fish, of course, when we first anesthetize them shortly, so they don't feel the pain. And then you can, within course of 1 to 2 hours, you can actually see the regeneration of the wound and see what happens in wild type, but also in fish with Myhre syndrome. And the model was already prepared by Michiel van Hooijdonk, who started on that more than a year ago with the funding of the Myhre Syndrome Foundation. Another thing we would like to pursue is 3D skin models. So for that, we will seed fibroblasts or skin cells of the mid layer of the skin, so the lower layer of the skin. And on top of that, we will put epidermal skins or keratinocytes that are actually the upper layer of the skin. And as this is done, you can actually make an in vitro model for skin. And this is a section of such a skin model. And this will enable us to look at wound healing specifically in a skin model of the disease. Also, we will look at how the SMAD4 protein is behaving both in wild type and in Myhre syndrome. See what is happening with the protein. Is it staying too long in the cell? Is it not breaking down properly, and so forth? We will look at the binding factors and also at cellular phenotypes. What's the cell motility about? How easily proliferate the cells? What kind of small hormones do they make that influence other cells in the neighborhood, and so forth. Finally, short word also, you might know that we are also working on clinical work. There's also the work of Eva, but also of Tim van Damme. So we are specifically focusing on the adult phenotype in Myhre syndrome. And we have now included 21 adults that are previously unreported. We have done the literature review and all the data collection. We're currently drafting the paper. So that's something that is coming up soon. And then finally, we also looked at episignature, meaning that sometimes a genetic defect can change how specific genes are being used by adding or removing small molecules on the DNA. And for many syndromes, there is quite what we call an episign. So you can actually sequence that specifically and then see what changes occur on the DNA. And this may help to interpret results of genetic testing, or may also help to interpret the mechanisms of disease. And we now have an episign that is also clear for Myhre syndrome. We are sending more samples for confirmation, but this should also be ready over the next few weeks. So with that, I would like especially to thank all the people in my lab that have been working on the work I presented. So the aneurysm model was made by Michiel. Eva is now working on the Myhre syndrome model. Tim did the clinical work. And then we have a whole bunch of collaborators, especially Patrick Sips, who is one of my major collaborators on the Zebrafish work that we do in our lab. Thank you so much.
Thank you, Bert. That's absolutely amazing to hear all of those updates. I've got a few questions for you, if that's okay, and we have some time. Sure. So you mentioned there a lot about phenotypes. Now, obviously, a lot of people on this call might know exactly what those are, but some people on the call might not know. So can you just give us a really basic explanation of what a phenotype is and what example you'd be looking for Myhre?
So what we mean by a phenotype is just symptoms of disease. So we know in Myhre, for instance, people with Myhre syndrome tend to be smaller than their peers. They may have neurodiverse development, so different than in other children. They can have some facial characteristics that are recognizable. And they also have this particular finding that there is a risk for fibrosis. So due to trauma or to surgery, that wound healing might be a little bit impaired and cause redundant tissue formation, which is called fibrosis. And so these are what we call phenotypes that we will specifically also look for in the zebrafish. So we can very easily see the vasculature and the heart development in zebrafish. Sometimes we see cardiovascular features in Myhre syndrome patients as well. So we can easily evaluate that in the fish. We can also look at fibrosis by this wound healing experiments. We also have a whole battery of neurobehavioral tests that we can use on zebrafish. So we can see how they interact with other fish in their tanks. We can also see how easily that it is for him to learn something new. So these are all things that we can easily assess in the fish and that we are very much intrigued by to see what will be there in the fish and how we can identify the mechanisms underlying such phenotypes and eventually, hopefully, identifying medications or therapy that may ameliorate some of these problems.
Mm-hmm. And so how does the phenotype have to be characterized in X percentage so that therefore you go, this is a classic phenotype of this in the cell structure, and then how do you then move that forward in terms of then testing it essentially?
It's actually important that you always evaluate multiple fish. So you have to have sufficient observations in order to do statistics. And that's also one of the benefits for zebrafish research. We have hundreds of fish that we can evaluate. Of course, you will also see some variation in the fish, and that's also what we see in humans with a certain disease. There is some variation over there, and the same is true in the fish. So we try to somehow quantify these phenotypes, so that in case we treat the fish, that we can see amelioration in numbers. So less fish with, for instance, a congenital heart defect or a vasculature problem. So that is actually how we do it. We try to find quantifiable parameters that we can easily assess in a large amount of fish. It's certainly not so that every fish should have the full phenotype of what we expect, of what we think of what Myhre syndrome is. We can also look at specific parts of the phenotype. For instance, when we look specifically at wound healing, we can do this also in a large number of fish. But it might be possible, for instance, that these fish have normal length so that they are not smaller than their wild type peers, but they do have fibrotic problems. So yes, that's something we will have to evaluate, and we will have to identify quantifiable parameters to kind of describe the phenotype in the fish, and then hopefully with medication, with drugs, we can kind of interfere with the process and bring it closer to the wild type fish.
Yeah. Questions here coming in. Would it be possible to do CRISPR-Cas therapy in Myhre syndrome patients in the future?
Yes, so that's of course a very relevant question. And that is, of course, something that lives strongly in the rare disease community. Theoretically, it would be possible, but there are still a lot of huge major burdens that are in between. For instance, it's at this point it's very difficult to actually target specific cells with CRISPR therapy. So it's not about we can inject this in the blood and it finds its way to the cell. Now you actually have to get it into the cell, and you will have to get it into specific cells. One of the cells you want to target would be neurons. Other cells that you want to target might be fibroblasts, and so you have to get it into these cells specifically. Another major problem is also that it's not yet specific enough. So in the fish, it's easy, you inject it in the one cell stage, and then you have hundreds of fish, and you pick out the one that have the right change. But if we are honest, we actually only see the right change in about 2 to 4% of our fish. So meaning that, in the majority of the cells, it just doesn't work properly. So that's these are still major burdens that prevent it from use, from clinical use. Other things that we are thinking about is because it's a specific variant that occurs in Myhre syndrome, is actually to target a specific gene that has the variant in order to only have one copy of the SMAD4 gene then. But we know that people with one copy of a SMAD4 gene don't have Myhre syndrome. They do have an increased risk for cancer, but that's something that is more manageable.
Okay.
But that's still also something that is not yet for clinical use at this point.
Right. And the really wonderful examples you showed on your slide deck of what the zebrafish model can show you, how far away are we from that with the Myhre model currently?
With Myhre model, we are very close now, because we do have the adult fish that are growing in the lab. So we can actually start, we will need to backcross them once more. So we can actually start and phenotype these fish from now on. So all the offspring, we will look at the vasculature and we will look at growth. These are the most easy parameters to evaluate, and then in the adult fish we will look at fibrosis in first instance. So that's something that we can do now over the next year. Of course, it's something that needs repetition. That's not always very easy to do, so it's not that the fish are there that we will have the results within 2 weeks. That's not how it works, but I think over the next year we should be able to get some results. Yeah.
Okay, and a final question. Has anything surprised you? Because we had Dr. Mark came on earlier and talked about the mouse model, and there's obviously another mouse model in France. With the zebrafish, has anything surprised you about your findings? Have they behaved in a different way to all the other zebrafish models you have?
Not for now, but we are still working now on a characterization. So we are still in the very early phase. So they tend to swim around normally, but that's all I can tell at this point. So we will see how it evolves over the next year. But I guess there must be some things that we will able to see in these fish. Yeah.
Thank you very much. No, that sounds amazing, and thank you to your team who have joined on the call as well, and for all the hard work that you guys have been doing. And thank you for joining us from Belgium, which I think now is Saturday evening.